This method is successfully optimized to classify breast cancer patients into known and novel therapeutic groups. The high sensitivity of this assay identifies heterogeneous samples that can be further studied using laser microdissection.
The main advantage of the RNA-based multiplex assay is the reproducibility of results using hematoxylin and eosin-stained formalin-fixed paraffin-embedded archival material, allowing high-resolution outputs and biomarker validation.
Prepare the tissue as described in the text protocol. To begin the dissection, set the slide limits and calibrate the stage movement of the microscope. Now scan the stained membrane slides using the 4x objective. Select and encircle the target areas for microdissection on the membrane slide. Keep track of the total area. At least 42 square millimeters is needed for adequate downstream RNA analysis. Then initiate the laser cutting at the defined parameters. Next, use the cap lift mechanism to retrieve the dissected section, and set the section onto the diffuser cap of a labeled tube attached to a mechanical appendage. If the dissected section is not retrieved onto the cap, repeat the dissection of the target area.
Accuracy of microdissection is a critical step, and one should always ensure that the microdissected area present on the cap coincides with the marked scan.